right, so this is, I've made a drawing here sort of representing meiosis. So meiosis is cellular division for the purpose of conducting sexual reproduction. So during meiosis, you produce daughter cells just like you do in mitosis, except for in meiosis, you produce daughter cells that are haploid instead of diploid. So you're cutting the number of chromosomes in half per cell. So in humans, you start off with 46 cells. I got 46 chromosomes in each cell. My skin cells are making more copies of themselves through mitosis, making cells with 46 chromosomes and 46 chromosomes and so on. In my testes, I'm going to make sperm cells, and I only want those to have 23 chromosomes because when they meet up with an egg cell, 23 plus 23 returns you back to your 46 chromosome number. So here we go. The top row up here represents the first meiotic division, or meiosis I. So just meiosis I, you can call this whole top row that. And you can see I've written a 1, basically Roman numeral 1, after each of these. So if you say anaphase 1, everybody knows what you're talking about. You're talking about anaphase of meiosis I. Prior to all this, the DNA is replicated. That's A and P1. We're not going to go back into it, but basically you've doubled the amount of DNA per chromosome, which is why you have these chromosomes that are X-shaped right here. So you have X-shaped chromosomes. So they've got twice the DNA. Now during prophase I, this is the most complicated step, and I've kind of really simplified it here. But I want you to see that I'm going to have what I'm representing with a black chromosome and one that's represented by a red chromosome. Well, the black chromosome represents a chromosome that I inherited from either my father or my mother, and the red chromosome represents a chromosome that I've inherited from the other. So I've got one paternal and one maternal chromosome. During prophase I, these two kind of get together, right? They'll come together and they'll exchange parts. So I've kind of drawn that squiggly red and black line here to represent that they are coming together. They're making a contact. That process of them coming together is called synapsis. synapsis. Not sure if you have to know that one for in here, but I'm letting you know it anyway. The chromosomes touch at places that are called chiasma, or chiasmata. So this contact location is a chiasma. So that's contact location. The whole deal here, this whole group of chromosomes, these two chromosomes that have come together, are called together a tetrad. Tetra is 4, so tetrad means you have 4 copies of the DNA. And if you wanted to look here, 1, 2, 3, 4 copies, right? The whole process, this is a complicated, this is why I said prophase I is complicated. The process is called crossing over, and that involves exchange of genetic information. So this is a whole process meant to recombine genes. You want to mix up your chromosomes. One of the great advantages of sexual reproduction is that you don't make clones of yourself. You make a variety of different types of offspring, which, when you look into the whole species, means that you're going to have a whole myriad of different phenotypes, different traits to approach the world with, right? So if you make a bunch of clones, that's great if the world never changes. If the world changes, let's say you get a huge virus that comes through and shuts down the whole world, then some people are going to be, because of the variation, some people are going to be more naturally resistant to that kind of thing than others, simply due to this improved genetic variation brought about by sexual reproduction. So what we've done in prophase I is we've dissolved the nucleus. This dotted line represents the nucleus dissolving. The chromosomes have met here in a process called crossing over, and they're exchanging DNA right here. You're going to see also that there are these little fibers here, and these are what are called spindle fibers. And the spindle is this whole thing. And I've only drawn a chromosome cell with 4 chromosomes because I'm not going to draw 46. So, but you can see that the spindle fibers have attached, and they attach to the chromosome here, and they're going to ultimately pull it apart. Okay, because you're going to want to divide the cell, and when you divide the cell, each of the daughter cells needs DNA. So prophase I, nucleus dissolves, chromosomes condense. That means they become sort of visible chromosomes. Crossing over occurs with all that contains, and you've got the formation of that spindle. We're going to skip right to metaphase. Now this is a process that doesn't go and teleport here. It's going to slowly, merge into this next phase, which is called metaphase. Metaphase, if you want to imagine that there's a line dividing this cell in two. They call this line right here, you can draw it any way you want, but in this case I've got to draw it up and down. It's called either the metaphase plate or the equator, okay? I usually call it an equator, but it's a imaginary line that forms between the two poles of the cell during division. Now what you can see is that the chromosomes have lined up in pairs on this metaphase plate. What I've tried to draw here, and which I gave up on right away because it's hard to draw, is that I've drawn these chromosomes having some black and some red ink, because that's kind of that reflects the fact that you've exchanged parts between these chromosomes. So you don't have a paternal and a maternal chromosome anymore. You've got two chromosomes that are sort of combined from the maternal and paternal chromosomes. You're really mixing up the genes. Chromosomes are lined up in pairs. If I wanted to draw 46 total chromosomes, I'd line them up like this: 2 by 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 2, right? But suffice to say, we're just going to stick with 4. During anaphase, you see that these two chromosomes, these homologs or homologous chromosomes, homologous in this case means that they contain the same genes. These homologous chromosomes are being pulled apart. That's the only big deal here in anaphase. So these phases are a lot easier. Metaphase, they line up in pairs. Metaphase 1, I should say, line up in pairs. Anaphase 1, they begin to get pulled apart towards the poles. Telophase 1, you can see the key component here is that you form this thing right here, which is called a cleavage furrow. Cleavage in this case is to chop. And the cleavage furrow ultimately will pinch these guys in half like that, forming 2 daughter cells. Well, let's count the number of chromosomes. Over here, in this particular example, we've got 1, 2, 3, 4 chromosomes. I've kind of already drawn them together, but there's 4 chromosomes there. In my testes, that would be 46. Over the course of meiosis I, I put half of the chromosomes on one side, half of the other chromosomes on the other side, the other half I should say, and divide it in half. So 4 to start with, and now we've got daughter cells with 2 and 2, which gives you the overall purpose of meiosis I, and that is to reduce the chromosome number. This is called the reduction division. We go from 46 to 23, or 4 to 2 in each cell. So meiosis I, it's a really unique division. You end up going from a diploid state, and this is something you'll need to know. Diploid just means you've got 2 copies of each chromosome, to a haploid state, which means you've only got 1 copy of each chromosome. So we represent that by using this symbolism. 2n, that's diploid. 2 times n is just simple algebra. And then over here, each of these guys is n, right? So n just is a placeholder to represent the number for your particular species. For us, n is 23. 2n is 2 times n, 46. Now this second row here, I've kind of shrunk a little bit because it's not as interesting, but this is meiosis II, and you see I've got P2, prophase II, metaphase II, anaphase II, telophase II. I wrote this times 2 because we've got 2 cells, right? There's 2 daughter cells after meiosis I. I just didn't want to draw it twice. So just this happens in 2 double. Prophase I, nuclear envelope dissolves again. The chromosomes condense. Here we have, you see our haploid number of chromosomes. Metaphase II, I hope I said prophase II, but prophase II. Metaphase II here, here's that equator again. But now you see that the chromosomes aren't lined up side by side, they're lined up single file. So if you see a single file line, you're dealing with either mitosis, which we've dealt with in the previous semester, or the second phase, the second division of meiosis, meiosis II. So that's why you see metaphase II. Anaphase II, now they're being pulled apart again, but you see the difference? This was entire replicated chromosomes being pulled in 2 different directions, whereas down here, you've got halves of a chromosome. But let's just represent this X with this hand, right? So here's the middle. This is called the centromere, the place where it's held together. And this is what you call a replicated chromosome. Now, each of these halves is called a sister chromatid. I know there's a lot of terminology here, but each of these guys, I'm going to draw it in right here. So this one and this one, each of those these are basically identical. These are called sister chromatids. I hope you can read that. Those sister chromatids are pulled apart in anaphase II. When they're pulled apart, they're no longer called sister chromatids. Now they're each their own chromosome. So two sister chromatids forming a replicated chromosome. Now we've got two unreplicated chromosomes. Each of these chromosomes has all of the information that you need on that chromosome to function as whatever organism this is. And then we get to telophase II, where we see a cleavage furrow forming again, and you split. So what would happen here if I drew this in times 2 is that we're going to get a total possible of 4 cells, 1, 2, 3, 4, from this original cell. Count the chromosomes. There's 2, right? This is a haploid cell. Each of these cells now has 1, 2 chromosomes. So each of these is haploid as well. Never mind that this is a unreplicated chromosome and these are replicated chromosomes. Like I said, prior to this, we doubled the stuff. You doubled the amount of identical information you have. So I basically made a photocopy of my DNA. That means that either cell can use it equally well. You don't have to have both of them. You don't even use both of them. That's the end of meiosis II, and meiosis II is just to make sure you've got only one copy of all the DNA in each cell, and it also doubles the number of cells, which you'll see is useful, especially when it comes to sperm cells. So I'm going to kind of roll over here. I think I can just roll right through on this. I'm going to roll over here to 2 specific examples of meiosis in animals and humans. So here we have on the left, so right here, you'll see that I've labeled it over here. Most of it is called spermatogenesis. Well, spermatogenesis is just meiosis in a male. Spermatogenesis is meiosis, and it makes cells called sperm cells or spermatids. So let's look at some of the stages here. Well, starting off, and this is occurring in your seminiferous tubules, in those little round structures that you found, that we saw in the testes sections. There are cells that line the outside of this thing. Here's a seminiferous tubule. So you might find little cells out here called spermatogonia. And a spermatogonium is just a sort of a sperm stem cell. It's kind of like a precursor to the cells that become sperm. But they divide mitotically. They're going to divide mitotically, making more spermatogonia, and they're going to also make cells mitotically called primary spermatocytes. So a primary spermatocyte is kind of like a graduate of the spermatogonium program, right? It's gone on, and now it's going to go through meiosis. This way you have, in males, you have pretty much an unlimited supply of sperm cells because those spermatogonia are just pumping out themselves and pumping out primary spermatocytes mitotically. So these guys are diploid cells producing diploid cells, as you remember from mitosis. Here I've condensed meiosis I into this. I'm not going to write prophase, metaphase, anaphase. I'm not going to draw all that. Prophase, metaphase, anaphase, telophase, and cytokinesis, which I may not have mentioned, but that technical process of splitting is called cytokinesis. I don't care if you want to consider it part of meiosis of the division or not. Just know that when the cell splits, it's called cytokinesis. Prophase, metaphase, anaphase, telophase, boom. Now we've got 2 cells, and this is these 2 over here. I don't know if you can still see that, but there they are. These guys are called secondary spermatocytes. Primary spermatocytes go through meiosis I to produce secondary spermatocytes. Secondary spermatocytes go through meiosis II, that's the bottom row over here on the left, to produce spermatids. Spermatids go through several phases. They have to mature. So when they're first formed, they don't have any tails. Then tails are added, and they add some more stuff. They add some enzymatic function. And that process of maturation of the spermatids is called spermiogenesis. So spermato is meiosis. Spermio is a maturation process. right? Addition of the tail and building up of the sperm, giving it motility. And this the spermatogenesis happens exclusively in the seminiferous tubules. Spermiogenesis starts in the seminiferous tubules and then is completed in the epididymis. At the end of this, you've got 1, 2, 3, 4 spermatids or spermatozoa or sperm cells for each primary spermatocyte you started off with, right? And males are doing this all day, every day, all night, every night. You're making sperm cells from the moment you hit puberty straight through to the moment you die, right? Now it drops off towards the end, but still giving it the old college try. Here we have females, right? And this is another process. It's meiosis again. This is meiosis, but its female version is called oogenesis. So it looks like oogenesis, but it's oo. You say oogenesis. Females are a little bit more complicated in a lot of ways, but here especially, meiosis isn't quite so even, as you can see. Oogenesis, females have a real, it's a real strategy that differs between males and females. Males choose quantity and females choose quality. So females are investing in one large cell that has lots of organelles, it has lots of resources, it's really massive, it's millions of times bigger than a sperm cell. Males produce millions of sperm, right? And just try for, hopefully you get some quality in numbers there. But females are really going to be more conservative about this because they're the ones, especially in very large animals like humans, and especially mammals, that are risking the most. Females have to, in humans, they have to carry the offspring for 9 months. They're the only ones that can feed the offspring with their mammary glands, at least evolutionarily speaking. They have the most health risk involved with the process, so they need to be they invest very heavily in this. So oogenesis looks different. We still have oogonium. You can see the comparative cells over here. Oogonium, these divide, the oogonia divide into mitotically again, into primary oocytes. These primary oocytes undergo meiosis I. I'm not going to draw it in again. This is meiosis I right in here. Meiosis I is that reduction division, so we're going to get two cells. A secondary oocyte and a little reject called a polar body. Now the polar body has DNA. It still has it has the DNA from those chromosomes, but it doesn't get any of the other good stuff. It doesn't get any of not much cytoplasm, none of the organelles. Basically, it's a cast-off because you are concentrating on this one cell. If you divided them and divided them and divided them, you'd have lesser quality cells. So just throw that one away. This secondary oocyte is the cell that gets ovulated. So when a female goes through puberty and starts ovulating and menstruating, the cell that gets ovulated once every 28 days is a secondary oocyte. You may have also heard, kind of backtracking a little bit, you may have also heard that females have all of the eggs that they'll ever have by the time they're born. This is a common thing that people hear. Nope, they don't have any of the eggs. It depends on what you want to call an egg. They've got all of the primary oocytes they'll ever have. So by the time a female's born, she's got thousands and thousands, not billions and billions of sperm cells like the male will actually end up producing, but thousands and thousands of primary oocytes, which bide their time until, for 12 or 13 or whatever years, until that female matures. Then she starts putting them one by one through meiosis I, becoming secondary oocytes and being ovulated. Now, when they're ovulated, they're not ready to be turned into an embryo yet. They are ovulated and still need to go through their second meiotic division. However, the signal for them to go through the second meiotic division is meeting a nice young man in the oviduct, right? So there's a sperm cell which introduces itself through the proper channels, meets, relatives, and then there's what they call sperm penetration. So the sperm penetrates this oocyte, this secondary oocyte, which immediately induces the second meiotic division. So sperm cell penetrates, the egg cell really kicks up its, the oocyte I should say, kicks up its metabolism dramatically, does a bunch of stuff like prevent more sperm from getting in, but it also undergoes this second meiotic division, meiosis II, prophase, metaphase, anaphase, telophase 2, and makes another polar body. This polar body might occasionally actually divide as well, so you could have up to 3 polar bodies. Doesn't really matter, they're thrown out. This is the important one. This is now an ovum. And what I've attempted to reflect here is that the female had her own nucleus right here. This nucleus has N chromosomes in it. The male has a nucleus inside the sperm cell with N chromosomes. N times 2, when they meet, makes 2N. So this united cell, the cell that's formed from the haploid gamete of a female meeting up with the haploid gamete of a male, once those nuclei fuse, forming a diploid nucleus, diploid cell, that's when you call it the first stage of an embryo, which is called a zygote, or sometimes you'll hear called a fertilized egg. That's all I got.